Welcome back for more AP Biology. We're going to be dealing with the ramifications of having cell membranes, which is controlling osmosis and diffusion. So we're going to look at how do we control osmosis and diffusion, or the movement of substances into and out of a cell. It's one of the things that the College Board likes to ask questions about, and there's a bit that goes in the book. So the first thing that we need to keep in mind is a little bit of physics that you don't know. And that is a phenomenon called the kinetic molecular theory, which is a nice way of saying everything moves. Not only does everything move, it moves with a certain amount of energy, so there's a bit of punch that comes with everything that moves. And we don't know how things are moving. Things move at random. We don't really know exactly what's going to happen next. We can see the patterns, but we don't really know where you go next. Diffusion is this random motion along what we would call a free energy gradient, or along some type of shift. The word gradient means a change. It's like a slope. Slope is a gradient. So as long as I move from what we would call high free energy to low free energy, whatever free energy is, that's going to be diffusion. Typically we view this as moving from high concentration to low concentration, kind of like in this picture where you have the drop, high concentration, you have the beaker or the flask filled with water, low concentration, and the dye slowly spreads out. For the sake of pointing it out, this whole free energy thing is actually from something called thermodynamics. Thermodynamics is the study of energy and how you can manipulate it. And there turned out to be 4 laws. So the first law was called the first law. And there's a total amount of energy. The second law says no matter what you do, we're going to be increasing chaos, or we're going to be converting that energy from something useful to something that is useless. We call that energy free energy. The third one, the 0th one, don't really apply. They're good if you're going to be like a physical chemist or something like that. But basically, the third one is if you can make something perfectly still, meaning it has no temperature, it also has no chaos. And the last one has to do with having things being in equilibrium. So how can we manipulate diffusion? Because diffusion is just the movement of something from one point to another, or at least the overall flow. There's several things that we can do. So we can deal with concentration. So things will diffuse from high concentration to low concentration. Things tend to move from a high temperature environment to a lower temperature environment because we increase the temperature, we increase the motion. What we tend to notice is if we have a larger surface area, well, things can move across surface area. So if we have more surface area, I have more things moving across. I can have sizes of particles. The smaller things are, the faster they will move. The bigger they are, the slower they move. And we can also start to manipulate with what are they moving through. So like will move through like, meaning things that like water, like salt, will diffuse easier through salt than it would through oil, because like dissolves like. Well, how does this play into the whole cell membrane thing that we've spent a couple of days on? That has to deal with having membrane proteins that can help with the diffusion across the cell membrane. Recall that as you look at this picture on the far left side, that certain things, non-polar molecules, things that are fat soluble, don't care. They can move right across the membrane, and we call that simple diffusion or passive transport. It just happens. Everything else that cannot break through that phospholipid bilayer because it's too big or it's not soluble, it needs help. And that's the job, in part, of the proteins found in the cell membrane. So that will happen in a few versions. There's actually 2 different versions. One of them is you have something that can open and close, like a gate. And we call those gated proteins or carrier proteins. But you could also have things that are just holes. It's just a protein hole, and we call those channels. And those will allow for substances just to move back and forth ad nauseum. For the sake of pointing it out, not anything can move through anything, meaning if you have a channel that's meant for sodium ions, it will not allow potassium ions to pass through. It will not allow lithium ions to pass through. It will not allow calcium ions to move through. Anything other than sodium is verboten. It's not allowed. Similarly, potassium, which turns to be bigger than sodium, a potassium gate or a potassium channel will only allow potassium to move. It will not allow sodium to move, even though it's smaller. It won't fit. Proteins are picky. And if I do things to warp their environments, I end up warping the protein. So it's why, in part, cells are very nitpicky about maintaining their shape or their internal composition. We can describe diffusion for all sorts of things, but the one that gets a little tricky is when we start talking about water. And what we say is water doesn't diffuse from high concentration to low concentration, or from high pressure to low pressure, or from high temperature to low temperature. What we say is water goes from high to low water potential, because water has a whole bunch of things that can mess with it because it is the liquid that typically we dissolve other things into. So we need to take more things into consideration. Water potential is a simple way of viewing, or it's a holistic way of viewing concentration in a very strange sense, even though that's not entirely accurate. How do things diffuse? High to low concentration, or high to low temperature, or something along those lines. Water will do the same, just with water potential. We can see the results of water potential by looking at cells. So if I notice that a cell, when I put it into a solution, maintains its shape, what we say is that solution is an isotonic solution. It has equal strength. So the tonicity, so the pulling or strength, it pulls water out just as much as that cell pulls water in. Okay, got it. If you put the cell into a solution that makes the cell start to swell up, so it becomes turgid or it bursts, it becomes lysed, what we would say is that solution is hypotonic, meaning the solution couldn't pull hard enough. The cell pulled more water in than the solution could pull out, and the result is the cell got bigger. It increased in mass or it exploded. The exact opposite of that would be if something is hypertonic. That would be where the solution pulls on the water too much and it pulls water out, which will cause either, if we're talking about a plant cell, we call it plasmolysis, or it causes a cell to crenate or shrivel up. So water leaves, the mass goes down, the cell shrinks. We can put all of this into math. The top equation is the equation for water potential. The funky trident thing is called psi, P-S-I. It's a Greek letter. And the way you read that top equation is the water potential is equal to the solute potential and the pressure potential and the gravitational pressure. Or potential, excuse me, not pressure, but potential. The entire thing gives units in pressure because we're dealing with things moving, and moving is best described actually through pressure. So there are going to be units, things like atmospheres or torrs or bars or pascals or kilopascals. So they're units that you're not used to hearing. But those are the units found in that equation. The water potential is going to be the grand total number. And water is going to move from the highest water potential to the lowest water potential. That second term, the solute potential, is actually a math equation, which is where it says where, then it says the solute potential is equal to negative I CRT. I is called the Van Hoff factor, which deals with how many particles it falls apart into, and we'll do math practice of that in class. C is the concentration of stuff inside, hence the solute potential. R is called the ideal gas constant, and it's a value, and it depends on what the pressure unit turns out to be. T is the what we would call absolute temperature, which is based upon a phenomenon called absolute zero. If you've taken chemistry, you know what I'm talking about. If you haven't taken chemistry, you don't know what I'm talking about. But you have to use temperature in Kelvin. So it's going to be whatever the temperature is in Celsius, and you add 273 to it. The psi g, so the gravitational potential, has to do with water column height. And that is the density of water, which is that funky P thing called rho, which is density, times the gravitational acceleration times the height of whatever the column is. For the most part, we're going to ignore the gravitational potential, and we're going to look at the solute potential and the pressure potential. And as long as I don't squeeze or pull a vacuum, I can ignore the pressure potential. So basically, water potential will be the solute potential. Why point this out? Because I'm going to be asking you to do this on a worksheet. So this thing here is a graph that shows how the mass changed. So if water flows out, the mass goes down. If the water flows in, mass goes up. The catch is not everything probably starts with the same mass. So if you lose 10 pounds but you start at 100 pounds, that's gaining 10%. You lost 10%. But if you started at 200 pounds, you lost 5%. So what do you compare? Do you say that you lost 10 pounds, or do you say you lost 5% or 10%? The answer is we use percent change, which is why the vertical axis, the dependent axis, is change in potato weight, but it's being done so using decimals. So we're dealing with percentages. And then you have the independent axis, the horizontal axis, and that would be the solutions I put it into. What you can do from this is, if I were to make this plot and actually find the line of best fit, assuming we have no leakage and other weird things going on, turns out where that trend line passes through the x-axis, the x-intercept, that would be the C value of your water potential, or the solute potential, equals negative ICRT, which means we now have a number that we can plug into an equation to get a water potential number. How do you figure this out? You need to do an experiment to get those data points in order to make that trend line, so we can calculate C, so we can figure out the solute potential, so we can figure out the water potential. Why do we care about any of this? Well, it turns out we spend a lot of our time trying to control water. Water is most of what we're made out of, and if you're running short or running too much on water, we need to correct this really fast, because it actually throws a lot of things off. We actually have an entire 2 organs in your body, we call them kidneys, where their job is to regulate water. But we're not the only thing that does that. So this is a type of single-celled organism. It's a eukaryotic cell, because you can see internal membranes. It's called a paramecium. And what it has is, as you can see on the screen with me doing little twisties, is it actually has a bag that can fill up and then contract. We call that a contractile vacuole, and what it does is this cell lives in a hypotonic environment, so water is constantly rushing into the cell. Well, what do you need to do if you're in a hypotonic environment? We need to get rid of that water. So it has a contractile vacuole to spit water out. We turn out to live in a hypertonic environment, so what we actually need to do is actually retain water, and we need to get rid of toxins as they build up. If we look at birds, birds are also in a hypertonic environment. So what seabirds will turn out to do, not land birds, but seabirds, because they're always surrounded by salt water, is rather than, like with us, we make urine to get rid of the toxic parts that are building up. They actually have glands that secrete salt, which is why if you look at seabirds' beaks, they have white crusts all over it, and that's because they're secreting salt. And it's because they need to maintain their osmotic balance, their water balance. Trees will do the exact same thing, just with them it looks a little bit more complicated. It's also going to deal with things that we call transpiration, and things called stomata, which are little openings that kind of look like that eyeball or that mouth. They're found on the other side of leaves. We're going to deal with all this stuff a lot later. You're going to be doing a lab on this, so all of this is quite relevant. What we're actually going to be doing is attempting to figure out how salty cells are. So we're going to see if we can figure out that isotonic value for cells. The catch is you get to bring in fruits and vegetables, and we're going to find out how salty they are. All of that is stuff, so diffusion and that osmosis junk, all of that is stuff that naturally occurs. We need no assistance whatsoever. That is not true for all types of transport. We actually spend an extraordinary amount of our energy performing something called active transport, where active transport involves us using energy to force motions of molecules. Either I can force molecules into the cell, or I could force molecules out of the cell. And the way you do that is you have a protein, molecule comes on in, it binds, we add energy, and the addition of energy forces the protein to change its shape. And when it changes its shape, whatever attached initially, so in this case sodium or potassium, says, nope, I don't want to be attached anymore, and then it's now forced inside of the cell. The result is we increase concentrations of sodium and potassium or hydrogen or chloride or glucose or whatever into certain parts. It's a way of ensuring that if you need sodium outside of cells and you don't need it inside of your cells, it's a way to ensure that you don't have sodium inside of your cells. You always have it outside of your cells. It's a part of using the membrane to keep the internal environment of the cell as it's supposed to be. But we could also do a bigger version of this, and it's referred to as bulk transport. Typically, we call this endocytosis or exocytosis. So endocytosis is when a cell engulfs something and drags it in. We saw that with endosymbiosis. But if you just went backwards and took the thing that you brought in and spit it out, we call that exocytosis. They're basically the same thing, just reverse the order. It's not normal, which means we need to use a lot of energy. Even within cells, we need assistance in moving stuff around because we like to control the motion. And we actually have components of the cell called the cytoskeleton that actually help move parts around. And in particular, they have vesicles, so little bitty bubbles, that can move along these various cytoskeletal elements. This is a lot of stuff. I'm aware it's a lot of stuff. But what's the point of all of this? We have a cell membrane that has proteins to help regulate what's allowed in and what's allowed out. We can control the rate by using those proteins, but there's other factors that go into play that we get no say over. We don't get a say as to how big they are. We don't get a say in the temperature. We don't get a say in concentration. Ultimately, that leads to how fast things can diffuse, either into the cell or out of the cell, whether they're things that we want, nutrients, or things we don't want, toxins, waste products. What that ultimately leads to is a limitation as to how big cells can be. And it leads to the third pattern of biology. So we had surface area tells you something about its function, and form dictates function. We have another pattern here that deals with geometry, and we will talk about that later in class.